Hey class, in this recording we're going to start talking about Chapter 3 material. As we're looking at Chapter 3, it's going to focus on cellular biology or cytology, the study of the cells. And when we think of cells, we would be remiss if we didn't talk about cell theory. As we think of cell theory, we need to emphasize all organisms. So if we're alive, we're an organism, and we're going to be composed of cells and the things made of by the cells, so cell products. And I emphasized this in Chapter 1. I'm going to emphasize it again. The cell is the simplest structural and functional unit of all life. If it's smaller than a cell, it's not alive. If it's something that goes inside of the cell, it's too small to be alive. If it's a cell or bigger, it can be living. Doesn't necessarily have to be living, but it may be living. As we think of an organism, so the organism we focus on in this class is the human being. If an organism's structure is determined by the cells, then the cells of the organism also are going to determine the functions of that organism. A key part of cell theory is that cells only come from pre-existing cells. We are not going to focus on abiogenesis, which is a term that means we have life coming from non-living things, the first cell. We don't have a really good working theory right now. We have some, but they aren't very satisfying. As we look at cells within a species have a lot of biochemical similarities, but across the spectrum of all species, there are some commonalities among all living cells. There are some common biochemical characteristics across the species spectrum of cells. As you look at cells in human anatomy and physiology, we have about 200 kinds of cells in our bodies, and they have many different kinds of shapes. For instance, if we're looking at epithelial cells, and we're looking at a squamous cell, that squamous cell is going to be flat and elongated. It's going to be kind of shaped like a pancake. If we look at a cell that's kind of boxy or spherical, that's typically going to be called a cuboidal cell. And bear in mind, these are two-dimensional images of three-dimensional structures. And if we look at this next one, we have thick elongated cells. Those are going to be referred to as columnar. Now, if we look at a columnar cell from a side view, it's going to be very elongated. But if I were to take this picture and rotate it 90 degrees, those columnar cells may look like circles as I look straight down at them. So depending on your view, maybe that same cell type could look very different. So one of the things you need to keep in mind when you're looking at microscope sections is what kind of slice do you have through the tissue. That's going to influence the view. Other common shapes include polygonal, where they're just randomly irregular shape. We could have stellate cells, which are shaped like a star. We could have spheroid or ovoid. Those are commonly referred to or commonly seen with leukocytes or white blood cells. Discoid is a term that I don't see very often used. Most of the time when I see this shape, and when I was taught this back in the day, it was a biconcave. Or the jelly donut shape. We could also have fusiform. A fusiform cell is going to be pointed on one end, bulge outward in the middle, and then come to a point in the middle, and it'll be rounded in nature. And then we could also have fibrous. Fibrous cells tend to be very, very long, thread-like cells, sometimes up to 30 centimeters long. As we think of human cell size, most human cells are too small for us to see with our naked eye. Usually the limit of human vision is going to be about 100 microns. So this 10 to 15 microns or micrometers is too small for us to see with our naked eye. Human egg cells are quite large, and if you have good eyes, you can see a human egg cell with your naked eye. Some nerve cells, particularly the axons of these motor neurons, can be quite long, over 1 meter in length. The key part or the thing to focus on with these axons is they're so skinny that these axons are not easily viewable without a microscope. As we look at cells, the limit on the size of the cell typically is going to be related to its surface area to volume ratio. If there's too much volume relative to the surface area, the cell has a hard time transporting nutrients from the outside to the inside efficiently and can't support itself. As we increase the diameter of a cell, the volume of the cell increases faster more than the surface area. Surface area is calculated with radius squared, and volume is calculated with a radius cubed. So volume increases much faster than surface area. As we look at cells, one of the things we need to do with cells is to study them under a microscope. When we think of light microscopy, and I have some light microscopy shown right here, light microscopy does a great job of showing the plasma membrane, nucleus, and cytoplasm. So it shows big structures within the cells, particularly the nucleus of the cell is a very large structure that we can easily see. We in this class spend a lot of time focusing on light microscopy. We do not do any electron microscopy though. So we aren't going to spend time looking at TEM, transmission electron microscopy, or SEM, scanning electron microscopy. Both of these are pretty cool. I remember having an opportunity to do these when I was an undergraduate. We are going to focus primarily on light microscopy in this two-course sequence. If you have any questions or comments about this recording, please feel free to post them in the class discussion board or shoot me an email. And as always, happy studies.